Hi everybody, welcome to microbiology. We're on week 9. I'm Professor Gustin, and we're going to be talking about microbial genetics. This might be the most challenging week of the semester, or definitely one of the most challenging weeks. We have some challenging topics in both lecture and lab. So let's get right on it. We have in lecture, we're going to be talking about microbial genetics. And what I try to do in this lecture is remind you of some foundational information about genes, about DNA and RNA and proteins, and please pay attention to that. If you don't have that in your memory by now, as a biologist and as a scientifically thinking person, this is a really important concept, even for nurses who, when you talk about doing gene therapy and genetic problems, there's a good foundation here that will get you hopefully clear on that. So it's once and for all, get that DNA to RNA protein thing straightened out. And we're going to build on that in relating that to microbial genetics in particular. So I highly recommend that you watch the videos rather than get all the information from the textbook. There's a lot of genetics information and it gets really complex quite quickly. And so if you watch the videos, you can be sure that you get right to the points that I'm trying to make in this lecture. So please watch the videos. There is a lecture assignment, just like all the other weeks, our adaptive learning assignment from McGraw-Hill. So once you've completed your study guide and you've reviewed it, try that lecture assignment. In lab, we're actually going to be quantifying bacteria. So one of the first things we're going to do in the lab is, or one of the second things, I guess, is I'm going to give you, we're going to look at a colony of E. coli, and you're going to figure out how many bacteria are in one colony. And so what I'm doing is setting you up to be able to use a technique where you can actually count millions of cells. And this is a technique that we actually use in the live lab, and we do it. In fact, the data that I'm showing you are data that were collected in the classroom. So what you will see is I have two things posted here that look like the same thing, but one's a Word document and one's a PDF. What I have noticed is sometimes when people print out the Word document, the pictures that I have in here don't line up correctly. So I highly recommend that you open this and download it and copy the PDF unless you're typing right in the document. You can use the Word document, but make sure your pictures look good. And you might even want to go back to the original electronic version to look at the pictures. So print out one of these and do the lab. Now, if you've got any issues with math, or it's just intimidating or it doesn't click very well, you might need some help. And the direction, the math I'm doing in here, I assume very little knowledge, so little prior knowledge. So you should be able to do these problems from the directions that I give you. But if you're having trouble, please contact me. And it says, we can talk by phone, online, or you can come to my office. But every semester, even in the live class, there's always a couple people in the class that it just isn't clicking. So take your time. Do not wait till the last minute to do lab this week, because it might take you a day or two to get these math problems figured out. And if you're doing it when you're rushed and frustrated, it's going to be very hard. So please put some time into these doing labs. So print out one or the other of these. There's 2 virtual labs and a lab quiz. So go for it. So this week it's kind of working hard, doing things ahead of time, being ready for a challenge, and reaching out if you need help. So meanwhile, have a great week. Bye-bye.